Yes, you're welcome to NX videos, and at this time allow me to present about transport across the membrane or membrane transport. That's what I want to present and make sure you follow. So we shall begin with a membrane and try to understand what is on the membrane so that we can know how transport takes place. So membranes have what we call channels, and some channels we call them the leakage channels, and then others we call them the gated channels. Leakage channels are always open and they always allow in specific ions to move across or to move or to pass through them. They don't allow in any molecule. And you find that big molecules that cannot pass through the membrane require channels. For example, have some molecules here, big molecules. They are being represented by blue, and then others being represented by green. Hope you can see them. We would expect that because the membrane or because of the fluidity of the membrane, these molecules would be in position to cross the membrane, but that's not the case. Even if we increase their concentration, you find that uh, they cannot cross the membrane. Even if you increase the speed at which these ions or these molecules move, they cannot cross or move across the membrane. So that's why we need channels. Let's see. If we have a leakage channel, that channel, can it allow in all molecules? No. It can only allow in the green to move across the membrane, but specifically from the side where we have a higher concentration, the side where we have a low concentration. However, the blue molecules cannot go through because these leakage channels are specific. And some examples we have the potassium channel, ion channel, we have the sodium ion channel. Let me try to put in the blue one, and you see now the blue can allow in only blue ions to go through it, whichever side, but it only allows in a specific ion to go through the membrane. Then we have, oh, before we go to the gated channels, find that the leakage channels, they are always open, and find that membranes, the common channels, we have the potassium ion channel and the sodium ion channel, but you find that membranes normally have many potassium leakage channels than sodium leakage channels. Then let's try to see how the gated channel works. I'll place it there, and I'll place this one also there. When they are closed, no ion can go through them, and these are also specific in nature. Let me try to open the green one. You find that it's only the green ions that can go through, the blue cannot. Let's try to open the blue one. You find that the blue can allow in the blue ions to go through, but the green cannot. So the gated channels and the leakage channels, they are specific in nature. However, you find that the gated channels only open or close when there is a change in the when there is a change in the cell or across the membrane. For example, if there is change in voltage across the membrane, then these gated channels can either open or close. So before we move to the next point, I want to tell you that membranes have both leakage channels and gated channels. However, you find that some channels for specific ions are more than other channels. Okay, now I want to take you to the actual membrane and we try to analyze it in depth. I have a voltmeter. This one measures voltage across the membrane, and you find that if there is any change in the positives across the membrane, the voltmeter has to change. So this part is the outside of a cell, and this part is the inside of the cell. And at rest, when the membrane is not tampered with, you find that it has a specific potential, which you call the resting membrane potential, which is being measured, or which can be measured by a voltmeter. But in case there is a change in the distribution of ions across the membrane, the voltage changes. That's what I want to begin with and show you how the potential difference changes. For example, if I have this positive and is moved inside, I guess you can see that the voltmeter starts changing towards the positive. Let me move more positives inside. Moves inside. Okay. 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 Let me try to move over this one. So the key point is that as more positives or positive ions cross the membrane into the inside, you need to know that the potential difference or the voltage goes or moves towards the positive. However, if the positive ions move out of the cell to the outside, let's check what happens. That is the first one. Look at the voltmeter on the side and see how it moves. I guess you can see. So when more positive ions move outside, the potential difference across the membrane becomes more negative. That is a key point which I want us all to get to know. So if there is a change in the distribution of ions, specifically the potassium ions and the sodium ions, therefore the potential difference also changes, that is across the membrane. When more cations move out of the cell to the outside, it becomes more negative. Yet when more positives or when more cations enter into a cell, the potential difference increases towards the positive. Okay, I guess that point is clear. Now, remember, a membrane has gated channels, has leakage channels, but it also has what we call the sodium potassium pump. This one is good at maintaining the resting membrane potential. However, it uses energy. So I want you to see what happens or how the sodium potassium pump does before we can go any further. I want you to see. So 3 sodium out, 2 potassium ions in, but a single molecule of ATP is used, and a single molecule is used to cause the change in the distribution of ions. 3 sodium out, 2 potassium in. And during that process, I want you to see that there is a change in the PD. There is a change in the PD. I don't know whether you are able to look at that. Let me try to reset, and then you see what exactly happens. Check the potential difference is at 0 right now, but now I want to see what is happening. 3 sodium, 2 potassium in. So slowly by slowly, you find that as more sodium ions are transferred out or pumped out and few potassium ions are pumped in, you find that the membrane becomes, or the potential difference across the membrane is more negative or becomes more negative. So we want to say that at rest, the potential difference across the membrane is always negative. And we find that the sodium potassium pump always moves ions or pumps ions against their concentration gradient. What do I mean? In other words, the side which has more ions is the one which receives more ions. Just like in this case, uh, we have sodium ions in the inside, very many of them, and very few outside. And you find that when there is distribution of ions, like in this case, the outside will have more of the sodium ions. It will have more of the sodium ions, sorry. Yeah, it will have more of the sodium ions. It to have more of the sodium ions. So that's what takes place. Now, even if the process continues, even if up here we have more of the sodium ions and here we have less, still this pump will continue to move these few sodium ions left to this other side which has more sodium, and that's why we are saying that it always uh, moves ions against their concentration gradient. I guess that one is clear. Now, a full membrane has leakage channels, has the sodium potassium pump. It also has the gated channels. It has it, all that. But why is it that the inside is more negative and the outside is more positive? The key point here is that the membrane has more of the leakage channels of potassium than the sodium. So what does this mean? It means that the membrane is more permeable to potassium ions than the sodium ions. Let me make it more clear. The membranes of cells have many potassium leakage channels compared to the sodium leakage channels. Remember, these channels don't close, they are always open, meaning that an ion whose channels are very many has higher chances of diffusing or moving across the membrane more easy, and hence will accumulate on one side than the other whose ion leakage channel are few. Like in this case, just look, we only have one leakage channel for sodium and we have four leakage channels of potassium. And check the potential difference, it is in negatives. Let's try to see what happens. Let's try to see what happens when we have these leakage channels. So the sodium will only pass through the sodium ion channel, but let's try to see the potassium. So very many potassium ions can go through different potassium leakage channels that are present on the membrane. So find that at the end of it all, more of the potassium ions move across the membrane, that is to the outside, than the sodium that are capable of coming back into or being linking back into the inside. Remember, I want you to recall that we have the sodium potassium pump, for it always pumps the sodium out, out, out. But now remember again that the membrane has more of the potassium, it has more of the potassium leakage channels. That means that even though two potassium ions are pumped into the inside the membrane, again there are higher chances that many potassium ions leave the inside to the outside, meaning that the inside remains with fewer potassium ions and few sodium ions get into the inside of the membrane. I guess you understand that. So that's what actually happens. So how does the cell function with the leakage channels and then also the sodium potassium pump working? That's what I want us to see. I want you to be observant and see what exactly happens. So the sodium potassium pump will always continue to move the sodium ions out, although they are capable of leaking back inside the cell. But the sodium potassium pump will continue pumping them out, pumping them out, pumping them out as it pumps also the potassium ions in. However, the potassium ions, more of the potassium ions move out of this membrane, and therefore at rest, I want to emphasize this, at rest, the potential difference across the membrane is always negative 70 millivoltage. So the sodium potassium pump will always continue to move or pump sodium ions out from the inside of the cell to the outside, although they are capable of coming back, but few come back. Few come back. That is, few sodium ions come back across the sodium leakage channel. However, the potassium ions that are pumped inside the cell by the sodium potassium pump, many leave via the leakage channels. That's why the inside is more negative. I guess you understand that. I guess you understand. So, how do gated channels affect the membrane potential? I want you to focus voltimeter, on the voltimeter, and then see. Remember, the gated channels only change or only open when there is a change in the potential difference of the membrane. Let's say there's change in voltage, maybe a membrane has been stimulated. That's when they can open. Or when there are specific chemicals, or when there is change in pressure, that's when the gated channels can open. So I just want you to see, if the gated channel opens, let's say the potassium gated leakage channel, it will only allow the potassium ions to move out. And when they move out, you can see that the PD becomes more negative, more negative. Remember, the normal PD is supposed to be negative 70, but now if more of the potassium ions move out, it goes below negative 70. Let's try to open the sodium gated leakage channel and we see what happens on the voltimeter. So as more of the sodium ions get into the inside of the cell, the membrane becomes more positive. So in that case, if it becomes more positive, we say that the membrane is becoming depolarized. It is becoming depolarized. In other words, the sodium ions change the polarity of the membrane. Originally, it was negative, but when they enter, so at this point here, let's say at negative 70 millivolts, the membrane is more negative in the inside and more positive on the outside. So when the positive ions start moving from the outside to the inside, it is no longer polarized, but is getting depolarized. That's why we talk of depolarization. So during depolarization of the membrane, it becomes more positive, or the potential difference starts rising from negative 70 millivoltage, negative 50 millivoltage, positive 30 millivoltage. So that's how it changes. However, when the potassium gated channels open, you find that potassium ions move out and the membrane becomes more negative. It becomes more negative. That's what happens. That's what happens. I guess you're able to follow the movement across the membrane. Now, let's have an application that we can discuss and see what happens, and that is using the axon. Using the axon. The axon is part of a neuron where impulses get transmitted, and that's where I want, oh, that's the application of this membrane transport that we want to use in this simple illustration. Uh, we have a simple neuron whose axon is being shown, and what you see here is that is the inside, that is the axoplasm. It contains ions such as sodium ions, potassium ions, chloride ions. We also have proteins. All those are ions. And I want to tell you that the charges are distributed in this way, that the inside is more negative. I guess you're able to see the inside. The inside is more negative, and the outside is more positive. Is more positive. So, and then we need to know that the concentration of the sodium ions is also different from the potassium ions. So when you look at the membrane that is at rest, we have more of the potassium ions inside than the sodium ions. And when you look at the outside, we have more of the sodium ions than the potassium ions. So On our membrane, we have the leakage channels. Hopefully you can see them. They're ever open. They allow molecules either to move out or to move in, to move out, to move in. However, we have the gated channels. We have the gated channels. Hopefully you can see them. Have the gated channels. They only open when there is change in voltage. And what can cause change in voltage? For example, arrival of an impulse on in the axon. So let's try to stimulate our neuron and we see how the gated channels open. Remember, we have gated channels, that is the sodium gated channels. We can also have the potassium gated channels. Let me try to make them more clear. The ones which are red, those are the sodium gated channels, and the ones which are green, those are the potassium gated channels. I want you to look at the difference or what happens when the impulse arrives. I want you to look at the distribution of charges when a neuron arrives. So the gates that open first, I'll pause there first. The gates that open first, they are the sodium gated channels before the opening of the potassium gated channels. I don't know that you were able to see that. Let me try to repeat it in a slow form so that you can see. I want you to look critically. I want you to look critically and see. So on the arrival of a neuron, on the arrival of a neuron, so on the arrival of an impulse, you find that the sodium gated channel opens first before the potassium gated channel open. I want you to observe clearly. Our impulse is coming. On the arrival, the sodium gated channel open first before the potassium gated channels open. So what does this cause? This means that the inside has an influx of the positive ions from the sodium ions, and therefore there is depolarization or immediate depolarization of the membrane. So the depolarization of the membrane at one point will cause opening of other gated or other sodium gated channels along the membrane, and therefore more sodium ions will get into the axon or inside the axoplasm, and this will mean that the inside becomes more positive and an impulse is immediately generated. However, in a fraction of a second or after some milliseconds, like 0.05 milliseconds, the potassium gated channels also open. And when they open, this means that now the potassium can diffuse outside, and therefore the membrane gets repolarized again. It gets repolarized again. So I want us to analyze the chart so that we can get to know what exactly takes place during the stimulation of a neuron. During the stimulation of a neuron. Okay, we are going to use this simple chart. We're going to use a simple chart here to see exactly what happens on stimulation of a membrane. So originally, or at the start, the membrane potential is negative 70 millivoltage, but I want us to see what exactly happens on the potential difference as the membrane is stimulated. I've stimulated it, check. Immediately on stimulation, there is a rise in the membrane potential. So along this point or at this point, that's when the sodium ions influx or get into the membrane. They get into the membrane and then into the cell, and therefore the inside has more positives than the outside. So that's why there is a change in the membrane potential. The potential here increases. So if we were to use our voltmeter, if I was to take you back to our voltmeter, you find that when the sodium ions enter, you find that the membrane becomes more positive. That's why on this neuron we are saying that there is a rise in the membrane potential. So it means that, or when you look at the graph and you see a rise, it indicates that there is an influx of the sodium ions inside before the opening of the potassium gated channels. Then at this point, you're seeing that now the membrane potential is going down. Why is it going down? It is because of the opening now of the potassium gated channels. When they open, now the potassium ions can diffuse out of the membrane. And given the fact that we have more of the potassium gated channels and more of the sodium leakage channels, more of the potassium ions diffuse out of the membrane. That's why there's that movement, and this movement down is what we call repolarization. However, if there is a delay in the closure of the potassium gated channels, then we lose more of the potassium ions, and therefore the cations, and the inside becomes more negative. So below negative 70 mil voltage, we say that there is hyper polarization as a result of delay of closure of the potassium gated channels. So, however, after some time, we are seeing that after a fraction of seconds, now the potassium gated channels close. And when they close, even the sodium gated channels close, and therefore the sodium potassium pump can now carry out its role of restoring the membrane potential back to norm. And then also we shall have the membrane potential being restored back to norm. That's what actually happens. That's what happens. So let's try to see the distribution of ions. So the distribution of ions will definitely change depending on which membrane has been, or which when the neuron is stimulated. So that's what, my friend, that's what happens. That's what happens. So the more the membrane is stimulated, the membrane potential will always change. And that's what happens during membrane transport. So in summary, let me try to summarize. Uh, During or at norm, or when there's no stimulation, the membrane potential is always at negative 70 millivoltage. However, on stimulation, the membrane potential starts rising up to around negative 55. Any further stimulation beyond that will cause the membrane potential to rise beyond this value. It will cause the firing of an impulse, and therefore more sodium ions will enter inside the axon to cause depolarization, which can lead to production of an impulse. So at this point, or at the peak, that's when we have maximum sodium ions inside the membrane. And at that point, the inside is more positive than the outside. And then still at the peak here, it is only the sodium ions that are, the sodium gated channels that are open, but the potassium gated channels are not yet open. However, when we start moving downwards, That's when the potassium gated channels are now open and allow more of the potassium ions to diffuse out. Hence, repolarization up to this point. However, because there is a delay of closure of the potassium gated channels, there is hyperpolarization, meaning that the inside becomes more negative than the resting membrane potential. That is caused due to the delay in the closure of the gated channels, and specifically the potassium gated channels. However, later on, after a fraction of seconds, they close, and then the sodium potassium pump tries to pump sodium ions out and potassium back into the membrane in order to restore the membrane potential back to norm. That's what happens during during membrane or during the transmission of an impulse. I guess you have enjoyed this video. I know you have enjoyed this video. uh don't forget to subscribe on my channel. Don't forget to share it with your friends. Yeah, thank you very much. Thank you. I guess you have enjoyed. Love you so much. Bye bye. Bye bye.